I told a joke about dPCR at the lab once, but it didn't amplify well.
Welcome to Absolute Genius, a podcast series from Thermo Fisher Scientific. I'm Christina Bowens.
And I'm Jordan Ruggieri, and today we have a special episode featuring Dr. Clarence Lee, a senior product manager at Thermo Fisher.
Clarence and I will be at Bioprocess International this week in Boston, presenting on workflow efficiencies using QuantStudio Absolute Q AutoRun dPCR suite. If you're not able to attend his presentation, this interview is going to be a really great opportunity to learn about his role in the lab and the use of digital PCR technology. We'll also be talking to Clarence about his role as a product manager and how his past bench experience helps him better serve his customers.
Well, Clarence, thank you very much for joining today's episode of the Absolute Genius. We're super thrilled to have you.
Well, thanks for having me. Super thrilled myself just to hang out with the two cool colleagues at Thermo Fisher. Proud to say that I've been part of Thermo Fisher for about 18 years now in a variety of roles. Really started off within R&D, but within those 18 years, I'm super happy to say that for the past few years, I am the senior product manager for our digital PCR business. So currently very focused on our assays and applications efforts within that role.
Yeah, that's where we actually first got to know each other, is way back when we got started with all of the dPCR. And you were actually one of the first people to start, building out our application spaces. And that could be a good place to get started, is talking about, where we really started on our applications and, where our first ventures were into dPCR.
That was very recent, actually. Feels a while ago at the same time. But I mean, Christina, yeah, you learned a lot from you, actually. And a lot of our assay efforts has been very focused within the cell and gene therapeutic space, as well as, of course, with liquid biopsy. So built out a menu of liquid biopsy assays as an example to really take into account the circulating tumor DNA and the size of that, and looking for that needle in a haystack, right, of the mutations that we want to see with across the cancers for treatment monitoring or just MRD and all of that stuff, minimal residual disease for people not familiar with that term. And then the side of things within the cell and gene therapy space is we just see a lot of activities and interest in terms of using the Absolute Q and digital PCR system to quantify things without the need for a standard curve as they start building out these therapeutics, ensuring that it's built properly and then making sure it has great efficacy when it's being used for clinical research efforts.
One of the spaces that we see digital PCR being used for really commonly is the cell and gene therapy space, right? We see a big boom in people looking to adopt digital PCR technology. So Clarence, can you talk about why we would see digital PCR being such a useful application and why it kind of parallels qPCR so well?
It's a great question. I think in terms of what has been developed within the cell and gene therapy space as they look to develop analytical tests, process development, ensuring that what they're building is actually potent. And at the end of the day, it's a biologic, meaning it's an extremely complicated process. Being able to map out that process and study and understand and ensuring that process across the hundreds and thousands of steps that I'm sure it takes is extremely valuable. A lot of the stuff that has been built out is with qPCR, but that is what's interesting with qPCR and measuring is that you need a standard curve. Everything is based off of a relative value across controls that are, let's say, well characterized or understood. But you can imagine if there's a potential discrepancy with that control, then how do you ensure that what you're measuring is true? And everything's measured off of, let's say, a ruler. All right, so I'm measuring off a ruler that's set at this particular dimensions from 0 to 15 centimeters. But if I didn't place that ruler at a correct spot in terms of what I'm measuring, then my ruler's shifting. So how do I measure that? Am I really sure that measurement's great? Now, the beauty of digital PCR is that you are literally measuring a molecule. Right? You know that there is statistically a single molecule, and you start amplifying it and measuring that. And you don't need a set of standards to be able to quantify it. It's called digital because you're measuring the signal itself and saying, is it there? Yes or no? And then you're just simply counting. And so that's really helps provide some confidence. I mean, ultimately, the confidence that you are calling it correctly. And as you think through that, it's a less complicated problem as well. You don't have to make sure the standards are set properly or prepared properly. You have just greater productivity and being able to really ensure that the process is sound when you're developing your cell and gene therapy efforts.
Yeah, and that's really cool for things like rare molecules that you're looking for as you're looking for products within the cell and gene therapy process. But that can also be super helpful when you're looking for novel markers too, right? You're not having to go find and source standards for novel targets. I can see that being a really big advantage as well.
When you know what you're looking for, then yes, it's definitely something that helps out. You don't need to waste time in real estate, let's say, to prepare a set of standards where, okay, I'm going to build a set of standards so I know what that needle looks like and how much it should be. And instead, I'm just going to, I'm going to see. I'm going to do my analysis and look for that biomarker that I know I'm interested in or a potential contaminant. In that sense, you're also trying to show, yeah, I do literally have a needle in the haystack. Of course, people are always concerned about the reproducibility of your results as you go from user to user. And here you're kind of taking away one variable, a very important variable when you think about it. Okay, how does that person interpret what that standard should be and where, like, how I should do it? And that could be within the lab or in between labs and stuff like that. And now it's like, well, we have a method where, everyone knows how, like, is there a signal or not? Yes or no? That's it. And so now there's a common agreement and you see the value of what digital PCR can bring.
So kind of on that same line, thinking about kind of removing variables, the absolute Q does it itself, right? Like we remove a lot of the variables out of the digital PCR process just by automating so much of it through the single plate, single instrument design. And I know one of the topics we wanted to talk about today was how that translates to higher throughput needs, right? So when you really start to scale up your operation and you need to turn to robots, I'll start kind of like grounding us back in applications. And Clarence, can you talk about, where are some applications where you see the need, some use cases where a group or a set of, users might turn to automation to really help them improve their workflow?
When you think of automation, sometimes people just think assembly line, right? Like put it through like building a car or whatever the case may be. In certain aspects, that can be true, but ultimately what you can enable with automation is just higher productivity, how to better use your time. Do I really want my super important graduate student or somebody important within biopharma spending your time loading an instrument? Probably not, right? I want them really applying their critical thinking skills. And so, Christine, as you've noted, that what's great about the absolute Q and how it's been built out is that I like to use the term, the plate is the machine. When you think about it, the plate and the cool innovation about the plate is really what drives a lot of the simplicity and ease of use that we have with the instrument. And so you can definitely have an automated setup, whether it's, hey, I just want to run a lot more samples overnight and then come to data so I can just start analyzing it and just keep on going. And so we see that with particular applications from an environmental testing standpoint or with, a core lab where you might have multiple users wanting to run things. And so they just set the plates and then just run it overnight and come back to data. The other beauty of the absolute Q is we think about the entire solution. It's not just the instrument, but our assays, our consumables, and ensuring that we have the capabilities where we can have automation, because you just want to kind of set it and forget it, for a lack of a better term. And so we do that as well. Now, to add to that, since you're asking, well, what about what other applications? Definitely from a high throughput automation. Like if you're just running hundreds of samples, and we get that frequently when just research within infectious disease, or again, going back into the cell and gene therapy where people are developing therapies with adeno-associated virus, and they just are looking at a variety of samples across the process, from, let's say, crude lysates to all the way to just the initial, the final purification process. So they understand the process. That's definitely other aspects that people are looking into.
So easy a robot can do it.
Well, I like to say so easy that Clarence can do it.
I love that it's super approachable automation, I think, because so much, I think you inadvertently are already doing so much of it automated just by using the absolute Q as a base. And then just the way that the automation can take over to just, and I think you have it right, enabling better productivity. It's very approachable for whatever scale that you need.
Clarence, maybe taking the absolute Q into consideration here, how do we enable automation? Outside of the plate and of the absolute Q, how else are we enabling automation with our product offering and in collaboration with our customers?
With Thermo Fisher, we joke, but we do literally have pretty much everything. And so we do work with a lab automation team with experts in terms of understanding how to automate from a sample storage to actually getting out the data and analyzing it appropriately. And so if there are needs to integrate the absolute Q within automation that we have set up, like a cold storage box, whatever the case may be, we have the capabilities to do that. The other capability, of course, as I kind of highlighted before, is just the amount of service and support we provide. So you may already have high throughput automation already enabled. You want to integrate it into solutions that you have. So, we have a service and support team that can point you in the right direction, and we clearly have software that helps enable that. And then the other aspect is with the absolute Q, kind of highlight this, but not really. We do have what is the new Thermo Fisher Absolute Q AutoRun digital PCR suite. And so as part of our efforts with our lab automation team, we have successfully built out a unique system with a Spinnaker microplate mover robot that helps scale, right? So now you don't need that big, huge, complete soup to nuts automation. We have a suite where we enable plate loading and also provide some guidance in terms of loading the plates with our liquid handler. You can kind of set it and forget it with this AutoRun system. With the Spinnaker microplate mover, we have hotels where you can just load up the plates and you just hit go with the software. And what's really nice about the software actually, and the one cool part about automation is that you're just going to get a wealth of data, right? Now, instead of a single plate of data, I'm getting hundreds of data sets now, hundreds of samples within my data set. And so the really cool and innovative part with the AutoRun and Absolute Q software now is that we're enabling the capability for the first time in digital PCR, analyzing hundreds of samples in a single data set.
Those tools are extremely valuable for being able to analyze big data sets. I know that is one challenge, especially when you look at qPCR, which is just known for its really, really high throughput capabilities, where you're able to just generate just massive amounts of data. And digital PCR data is a little bit different to look at, right? You're looking at lots of dots and thresholds and copies per microliter. So to be able to visualize all that together and be able to threshold, to group, and to analyze all of that data together can really give a little bit of extra confidence. I know it's been really helpful in kind of showcasing that to customers and showing them what their data looks like across plates and helping to showcase just how consistent and reproducible things look.
Did you know that the QuantStudio Absolute Q dPCR system is powered by Microfluidic array plate or MAP technology? This technology offers numerous advantages, such as consistent microchamber filling, uniform sample digitization, and efficient analysis of more than 95% of the loaded volume.
So when you're looking for a needle in the haystack, it's pretty important that you don't throw away all of the hay first, right?
Exactly. You can learn more at thermofisher.com/map16, that's MAP and the number 16, or visit the Absolute Genius webpage. The Applied Biosystems QuantStudio Absolute Q dPCR system is for research use only, not for use in diagnostic procedures. And now, back to our conversation.
One of the big applications that we've seen get a lot of traction, especially after the big movements with wastewater, is this environmental DNA testing. And one of the reasons why, we've seen a lot of popularity is because of this buzz around inhibitor tolerance in dPCR. So I think this would be a really nice opportunity, Clarence, to get your expertise as to, why we see this quote unquote inhibitor tolerance, because we know it's not that dPCR is immune to it. So why do we see this with digital PCR?
Oh, so that's a great question. So digital PCR definitely is more robust to PCR inhibitors. And so for people who may or may not be familiar, of course, as you're doing real-time PCR and you're doing an amplification of DNA or RNA, as the case may be, in a bulk volume, there might be potentially within environmental samples, and you can easily imagine wastewater, there might be a lot of things in there that would kind of stop and really make it hard to amplify DNA and RNA. And so the very cool thing about digital PCR in general is that now you're splitting up that bulk reactions into tens of thousands of micro reactions, a lot smaller volumes. And so now the probability of an inhibitor being in the same reaction as a single molecule that you're trying to amplify is a lot less, and hence you're going to have a greater probability and hence the robustness of being able to amplify even with inhibitors in place. So you definitely see people wanting to use digital PCR for just in general environmental testing and pathogen detection. And the very interesting thing, I don't want to say cool because we don't want to deal with a once in a century pandemic ever again. The silver lining in this is that it really showcased how environmental testing and wastewater testing can have such an impact in being proactive within just public health.
One other thing that I wanted to touch on too, just thinking about, better confidence and better tolerance to inhibitors is some of the features within MAP technology itself, right? We know that digital PCR, you just talked about how good it is at kind of like naturally diluting out inhibitors so that they have less of an impact on the final, reaction kinetics. But there's a lot baked into MAP technology that helps to further reduce the impact of false positives. Can you talk a little bit about that?
The very cool thing about the MAP technology and the innovation associated with that, I already highlighted earlier, the plate is a machine. And it's really true because of the innovations put into it. You don't worry about wasted sample because you're going to process most of, like 95% of the volume that you actually input in. Because the really cool part about the technology is actually, and if you've never seen the plate, there's only an input. There's no output, meaning that you push in things, it's a gas permeable plastic underneath so that all of the gas bubbles, everything else is pushed out. You're maintaining your volume.
Clarence, I have another question for you. Are there any considerations as you scale up from, maybe you only have a handful of samples to hundreds of samples when it comes to the number of micro reactions that you want to make sure that you're utilizing?
No, that's an interesting question, Jordan. I think to your point, if you've got a big giant haystack looking for that one needle, then you need to look at a little bit more volume. And what's nice about a plate format is that, and actually with the Absolute Q, is that you can do what we call digital pooling. You can load your sample across multiple arrays and then analyze all of those arrays as one single sample. So if you're really looking for that 1 in a million event, literally, you can. So one example would be like looking for BCR able, right? Like just looking for that particular biomarker, and they're looking at 0.0032% if I'm doing the math correctly in my head. And so you can do that using the plate technology, and you don't have to. What's nice also about that, again, getting back in terms of the consistency about just the data or generating those microreactors, you can be confident that, let's say you need 100,000 microreactors. All right, I'm just going to load up 5 different sample wells to get that 100,000. I don't have to worry about, oh, am I only going to get 80,000? Am I only going to get 60? The other thing that you kind of highlighted, like, okay, maybe what if you're not doing hundreds of samples? What if you're doing only 4 samples? The beauty of a plate technology, and actually with the Absolute Q, is we've put that into consideration. You don't have to use the whole plate at a single time. The plate is set up where you can actually provide like 4 samples, like using what we call our MAP16 plate as an example. The MAP16 can analyze 16 samples at a given time. You can load up a single column of samples, and then as long as those other columns haven't been used, the system's smart enough because there's a barcode so that you can reuse that plate. So if you have another 4 samples, you can reuse that plate. You're not wasting it. Just use that plate again and just reload the next 4 samples.
Yeah, there's a lot of flexibility. I like that you're kind of calling out like the 2 sides of flexibility there. There's the flexibility where you actually need more reactions per sample, and you're able to like digitally pool your data within a single well to artificially create like a larger well. And you've also kind of talked about earlier where we're talking about digitally pooling studies together, where you're actually able to achieve the sample per run throughput that you need. Without actually having to make a consumable selection based on, what your desired throughput need is. So you're really selecting, exactly what works for your experiment based on analysis post.
All right. Now we get to hop into the career corner part of the podcast, and it is Jordan's career corner now. So trumpet sounds, French horns, any brass instrument. That's right. So Clarence, and talking a little bit about your background, you mentioned you were at, went into NGS and you spent time in the lab and all of that. How did you go from working in the lab to a product management position where imagine it's more about market trends, products to develop, and even, the R&D aspect is not maybe necessarily the lab chemistry or developing assays, but tweaking software, right? Or tweaking hardware. Can you kind of bridge that gap from bench work to automation and projects, that you're currently working on from a market standpoint?
So in terms of my background, grew up in Canada, actually went to University of Western Ontario, but actually I'm a chemist by training. So I did my PhD in chemistry and biophysics. I pivoted to genetics and sequencing as part of my postdoc. To kind of highlight how old I am, it was because of the movie Gattaca, right? It's like this dystopian future of what happens when you do rapid sequencing. So that's how I got into doing a postdoc at UC Santa Cruz to work on nanopore sequencing. And decided I want to go into the business. So I actually took quite a few different roles on the business side. So I went from R&D to strategy and tech assessment to actual global marketing development, commercial operations, and now within product management. So that's how I approached it. And ultimately, from a career standpoint, what really helped is a couple things. One, just being proactive. And two, although I'm not very good at it, is just making connections and networking. I don't like social media. I'm not an influencer. I'm an old person. But definitely one of those.
There's more to connections than social media. We like to chat till late sometimes for you too.
We do, because again, we all, why the two of you are awesome. And like I said, a lot of the, working with Thermo Fisher is awesome. We just want to serve. And we want really, it sounds very cliche, but when we say that we want to enable our customers to make the world healthier, cleaner, and safer, it really is true.
That's really our job, is to make science easier, for everybody in the lab. So I echo that 100%. Can you elaborate a little bit on the day in the life of a product manager? And our audience is a lot of times in the lab. So what does it look like to be a product manager, and kind of what does the day-to-day encompass?
Oh, well, I think what brings me the greatest joy about product management is that every day is different. You're managing a portfolio of products, and your main goal, of course, is to increase revenue. You're part of a business. But to do that, you interact with people across functions. You work with marketing, commercial, operations and manufacturing, R&D. You're working across all of these different functions. There are certain days where you are interacting more with manufacturing, because maybe there was an issue with a manufacturing process and you're trying to address that. Or you want to work with commercial and operations because you see data that suggests, hey, if we focus in this particular area, we can address more customer needs and help generate revenue. The common thing, and why I love it so much, is because, again, I get to learn and something new every day from different people. Oh, the cool part also, I forgot the one cool thing about product management. I get to talk a lot with customers. I mean, that's the best part. I find a lot of joy in just hearing what they're doing. The one thing about me getting into science ultimately is that I just love science in general. So just hearing about all of the different things that they do, as an example. I'm going to geek out a little bit. So my PhD was on the lateral diffusion of peptide aggregates in model membrane systems. A very, very specific narrow topic. But now, by working as a product manager and supporting a whole bunch of customers across different segments, I learn about cell and gene therapy. I learn about liquid biopsy, non-invasive prenatal testing, food safety, done a lot of cool things that I never thought would have been possible.
Sounds fun. All right, my last question, it's a two-parter. We ask everybody this, so are you ready? What is your most embarrassing lab moment, and what is a moment that you're most proud of?
Oh, all right. The most embarrassing moment. Oh, this one you'll love. I can't believe I want to say this on the podcast. So my most embarrassing moment was during my PhD. I was actually working with a fluorescent probe and synthesizing nitrobenzodiazole. While I was getting it all prepped up, getting it all scooped up, I sneezed. You can see where this is going. So
don't sneeze in the lab.
This is me like trying to tear it appropriately, measure it appropriately from a chemical reaction. And just walking around, didn't realize, I thought it wasn't bad and whatever. And then the fellow crasher comes walking in, laughs her entire, like howling with laughter because I had my safety goggles on, but then all of this probe was all over the bottom half of my face, exploded all over me. But it's a fluorescent probe. It's not coming off. So I had this probe on my face for a few days. Suffice to say, I called in sick a couple of days.
Oh, that's amazing. And what about your proudest moment?
I made a small dent in the scientific community. So my one geeky thing, I'll say, non-invasive prenatal testing. So I was actually very fortunate to work with the people that basically invented it for next generation sequencing. And so I worked on a few papers and collaborations on that. Now, non-invasive prenatal testing is a standard, right? It's definitely used for people that women that hit a particular age where they might be at higher risk. And so my one kooky, geeky, proud moment was that when my wife was expecting our third child, the geeky part was because she had a higher risk, the physician and genetic counselor was like, oh, you should do some non-invasive prenatal testing. And so I geeked out on that just because, well, I played a part in that. So I have to say that was a proud moment.
Yeah, it's awesome. Clarence, thank you so much for joining us on this episode of The Absolute Genius. We really enjoyed our conversation with you. Thank you very much.
No, thank you for having me.
That was Dr. Clarence Lee, Senior Product Manager for DPCR at Thermo Fisher Scientific. We had a great time with Clarence, and we can't wait to share more great science with you in the future. If you're around BPI this week, stop by booth 206 and say hi to the whole DPCR team. Until then, stay curious. Like, I want to be there too. Come see me. Christina's also very cool. I'll be in the booth, hanging out, doing demos.